A protein that's basically notorious for triggering these really devastating, incurable brain diseases is apparently secretly moonlighting as a getaway driver for cancer cells.
Yeah, it's a massive functional contradiction.
Right. And that contradiction is completely at the center of this really fascinating 2026 research paper that we're looking at today. It was published in the journal Cell Death and Disease.
It's a great paper.
It really is.
Yeah.
So welcome to this deep dive, everyone. Our mission today is to thoroughly analyze this paper's investigation into a protein called Atx2, or Atx2 for short.
And if you follow neurology even a little bit, you probably already know about the human version of this protein. I mean, it's heavily implicated in things like spinocerebellar ataxia type 2, ALS, and, just a host of other really severe neurodegenerative conditions.
Yeah, but this source material does this huge pivot, like it completely steps away from the brain. Instead, it maps out how Atx2 operates as this sort of master amplitude dial for cellular life, death, and even metastatic invasion across the entire body.
We're really looking at a profound paradigm shift here regarding what this protein actually does at a baseline level. I mean, for decades, clinical research has naturally just fixated on the pathology, right? The way Atx2 aggregates in neurons and causes disease.
Because that's what we see in the patients.
Exactly. But this paper isolates its fundamental wild-type role. Like, what is it doing when it's not causing a disease? And it turns out it's an RNA-binding protein. The researchers demonstrate that Atx2 is actually this critical regulatory node in one of the most ancient, evolutionarily conserved signaling cascades in all of biology.
Which is the JNK pathway. Okay, yeah. Let's unpack this. Because to understand how Atx2 modulates everything from, early embryonic development all the way up to tumor progression, you first really need to dissect that JNK pathway.
Right. So JNK, which stands for C-Jun N-terminal kinase, is part of the MAP kinase signaling cascade.
Which sounds very technical, but it's basically the cell's emergency response protocol, right?
Yeah, that's exactly it. It reacts to heavy physiological stress. We're talking about things like UV radiation, inflammatory cytokines, or just really severe DNA damage.
So how does the alarm actually get sounded?
It's all based on a sequential phosphorylation relay. So a stressor binds to a receptor on the outside of the cell membrane. That activates a specific kinase enzyme. That enzyme then activates another one, which finally activates JNK.
It's like a cellular game of telephone.
Yeah, but a really urgent one. Because once active, JNK rushes into the nucleus and alters transcription factors, primarily one called C-Jun. This fundamentally changes the cell's gene expression. It shifts the cell's entire behavior in a matter of minutes.
And depending on the tissue and just how bad the stress is, JNK can do a couple of different things. It can trigger apoptosis, like programmed cell death to quarantine the damage, or it can initiate tissue remodeling.
Right. But when that pathway becomes chronically dysregulated, when it can't shut off,
it transitions from being this protective mechanism into a major driver of tumor progression.
Exactly. So we have this highly volatile JNK emergency system. And the puzzle this paper really solves is, where does Atx2 fit into that whole cascade?
Right, because we know ADAM2 is an RNA-binding protein, meaning it interacts with messenger RNA transcripts before they ever get translated into actual proteins.
It acts as a regulatory checkpoint. You have to remember, the JNK pathway is incredibly potent. It's almost too potent. A cell cannot afford accidental runaway activation of this thing.
It needs supervision.
Yes. ADNANG2 manages that threshold. When the researchers mapped out where ADNANG2 actually lives in the cell, they found it clusters heavily in these things called stress granules and P-bodies in the cytoplasm.
Which are basically dense little hubs of RNA regulation and storage.
Right.
Picture it like this. Think of your body as this bustling metropolis. If the JNK pathway is the city's emergency response system, like the fire trucks, the biological wrecking crews, then Atx2 is the 911 dispatcher.
Oh, I like that.
Right, because the dispatcher isn't physically putting out the fire, and they aren't swinging the wrecking ball, but they completely dictate the volume and the urgency of the response based on the info coming in.
That analogy actually scales perfectly to the cellular level.
Because if the dispatcher is hyperactive, you get wrecking crews just demolishing perfectly healthy infrastructure. But if the dispatcher, goes offline during a genuine crisis, the whole city burns down. The tissue collapses.
Precisely. Now, it was known that Atx2 played a role in the nervous system, but its job outside the brain was really this huge mystery. So to track exactly how this dispatcher functions, the researchers used a classic genetic model, Drosophila melanogaster,
The fruit fly.
the humble fruit fly. They are highly tractable genetic models, and their JNK cascade is actually remarkably homologous to ours. It's very similar. In flies, the primary trigger for this JNK emergency system is a ligand called IGR.
IGR, right, which is basically the invertebrate equivalent of human tumor necrosis factor, or TNF. Okay, so let's do a really quick structural recap for the listener before we dive into the fly experiments. First, we have JNK, that's our stress-activated signaling cascade.
Yeah.
Second, Atx2 is our RNA-binding dispatcher protein. And third, the researchers are using the IGR ligand in fruit flies to basically artificially pull the fire alarm.
Right.
So the immediate question this raises for you as the listener is, what actually happens to a living, breathing tissue when you artificially jam this dispatcher signal?
Well, the researchers answered that by taking the developing eye discs of fruit fly larvae and artificially upregulating that IGR ligand.
Just flooding the zone.
Flooding it.
Right.
And unsurprisingly, pushing all our IGR into the tissue pushed the JNK pathway into total overdrive.
And the result was just massive, widespread apoptosis, cell death everywhere.
Yeah, they looked at it under a microscope using a fluorescent marker called acridine orange. It specifically intercalates into the fragmented DNA of dying cells, and the eye discs were just glowing with apoptotic activity. When these flies eventually reached adulthood, they presented with what's called a severe small, rough eye phenotype. Basically, the tissue had auto-destructed during development.
Right. But the real breakthrough happened when they manipulated Atx2 in that exact same high-stress environment, right?
Yeah. They genetically knocked down Atx2 in those specific eye cells, meaning they used RNA interference to reduce the amount of the dispatcher protein available.
Even though the IGR stress signal was still blasting at full volume.
Yep. The fire alarm is still ringing, but without Atx2 there, the massive cell death was completely suppressed.
That is wild.
The signal just lacked the amplitude to trigger apoptosis. The eye tissue developed completely normally.
And then they did the reverse, right?
They did. When they overexpressed Atx2, pushed the levels way up alongside the IGR trigger, the apoptosis intensified dramatically, and they replicated this in the developing wing discs too. Forcing high levels of Atx2 in the wing tissue led to this runaway apoptosis, causing a distinct notched, reduced wing phenotype.
So basically, the dispatcher was artificially amplifying baseline stress signals into these lethal tissue-destroying commands.
Exactly.
Okay, so we've established that high Atx2 drives destructive apoptosis in normal wild-type tissue. But the paper doesn't just stop at healthy development. They applied this mechanism to a really aggressive cancer model in the flies.
Right. They used flies carrying the RAS-V12 LGL mutation.
Let's break those genetics down really quickly. RAS is this notorious oncogene. When it's mutated to the V12 variant, it essentially locks the cell into a state of hyperproliferation. It just won't stop dividing.
And LGL, which stands for lethal giant larva, is a tumor suppressor gene. It's responsible for maintaining cell polarity.
So keeping the cell oriented correctly.
Right. So when you knock out LGL while simultaneously activating RAS, you are creating the absolute perfect storm for metastasis. The cells are multiplying uncontrollably because of RAS, and they've lost their spatial orientation adhesion because they lack LGL.
It's a mess.
A total mess. In the fly model, these corrupted cells form massive tumors in the eye disc. And they exhibit this highly invasive behavior. They actually actively migrate down the optic stalk to invade the fly's ventral nerve cord.
Okay, but here is where I need to push back a little bit, because as I was reading this, we run into a glaring biological paradox.
Oh, absolutely.
When the researchers knocked down Atx2 in this specific Rasgrel tumor model, the tumors stopped invading. And the survival rate of the fly larvae, their pupation rate just skyrocketed.
Yep.
But wait, so in the normal wing and eye cells, turning up Atx2 causes them to undergo apoptosis and die. But in the tumor model, Atx2 is suddenly helping them grow and invade. How does that contradiction even work? How could it be an executioner in healthy tissue, but a survival driver for cancer?
That paradox is arguably the most important mechanism detailed in the entire paper, because it highlights the intensely context-dependent nature of the JNK cascade. You see, JNK is not just a binary switch for cell death.
It's not just on means die.
No, it's a signal amplifier, and it executes very different genetic programs depending on the cell's underlying mutational landscape. So, in a healthy cell, hyperactivated JNK triggers the caspase cascade. That leads to orderly self-destruction. The cell recognizes unmanageable stress and quarantines itself.
But in the RASL mutant, that apoptotic machinery is severely compromised. The cell's ability to, fall on its sword is completely jammed.
Precisely. Because apoptosis is blocked, that high-amplitude JNK signal has to be rerouted. It can't activate the caspases, so instead, JNK activates transcription factors that upregulate matrix metalloproteinases.
Which are enzymes that basically chew through the basement membrane.
Exactly. It also upregulates integrins for cell motility. The stress signal essentially panics the cell into a state of compensatory proliferation and invasive migration.
Wow.
So Atx2 is the volume dial. In a healthy cell, cranking up the dial plays the self-destruct program. But in a genetically corrupted cell, it plays the metastasis program.
Okay, that is incredibly elegant. Let's lock in another quick recap to keep all these mechanics straight. Bullet 1. Atx2 dictates the amplitude of the JNK response. Bullet 2. In healthy tissue, a high Atx2 amplified JNK signal forces apoptosis. Bullet 3. In corrupted cells, where the atectosis is jammed, that exact same amplified signal drives aggressive metastasis.
Spot on.
So, knowing that Atx2 is the volume dial is one thing.
Yeah.
But the critical question now shifts from the phenotype to the molecular physics. Like, how does Atx2 physically turn up the volume of the JNK pathway without actually interacting with the JNK proteins themselves?
Right, because it's an RNA binding protein. And the paper isolates this post-transcriptional mechanism with just remarkable precision. Atx2 achieves this control by targeting the messenger RNA of a kinase called HIP.
HIP, which is homeodomain interacting protein kinase. It's an upstream activator, right?
Yes, it's essentially the biochemical ignition switch for the entire JNK cascade. The more HIP protein you have present in the cytoplasm, the more JNK gets phosphorylated and activated.
Okay, so how do they prove Atx2 regulates this specific ignition switch?
They utilize a technique called RNA immunoprecipitation, or RIP. It's a really robust assay where you chemically cross-link RNA to its binding proteins inside a live cell.
You essentially freeze them together.
Exactly. Then you lyse the cell, break it open, and you use an antibody that's specific to Atx2. Usually, this antibody is attached to a tiny magnetic bead. You use that to literally pull the Atx2 protein right out of the cellular soup.
And because it's cross-linked, whatever RNA it was holding onto comes with it.
Yes. When you sequence the RNA that gets pulled down, you can identify the exact transcripts Atx2 is bound to.
And the RIP assay revealed that Atx2 heavily binds to the 3' UTR. The 3' untranslated region of the hipk mRNA.
Right. And for those who might need a refresher, every messenger RNA has a translated region, which is the actual genetic code for building the protein. But that code is flanked by untranslated regions.
The 3' UTR acts as the transcript's degradation timer, essentially.
That's a good way to put it. Under normal conditions, RNA-binding proteins and microRNAs bind to the 3' UTR, and they recruit these deadenylase complexes, like the CCR4 NOT complex.
And those complexes chew off the mRNA's protective poly A tail, which basically exposes the transcript to exonucleases that just shred it to pieces.
Right. It's the cell's cleanup crew. But by binding specifically to the 3' UTR of HIP, Atx2 induces steric hindrance.
Meaning it physically gets in the way.
Exactly. It occupies the regulatory real estate, physically blocking those degradation complexes from accessing the transcript.
Okay, so going back to our earlier analogy, if the mRNA is an instruction manual for building the HIP protein, Atx2 binding to the 3' UTR is like slipping a thick protective plastic sleeve over the manual's back cover.
Oh, I love that.
Because the cell's cleanup crew can't attach their shredding enzymes through the plastic, the manual survives in the cytoplasm significantly longer.
And because it survives longer, the ribosomes just keep reading it, they keep translating it, printing higher and higher concentrations of the HIP protein, which subsequently hyperactivates the JNK emergency alarm.
It's so cool.
It is. And the researchers visually validated this protective sleeve idea using GFP reporter assays in Drosophila S2 cells. They fused a green fluorescent protein sequence to the wild-type hipk 3' UTR.
So it would glow green if it was sticking around.
Right. And when they co-expressed Atx2 in those same cells, the fluorescence intensity just spiked dramatically. The protective mechanism is highly efficient.
And they also showed this isn't just about disease, right? Like, this mechanism is essential for normal biology.
Oh, absolutely. They demonstrated its necessity in normal embryogenesis by analyzing a process called thorax closure.
Right, thorax closure is this morphogenetic process
Yeah.
where epithelial sheets migrate and fuse to form the fly's dorsal midline. It requires really precise spatial coordination, which is all driven by baseline JNK signaling.
Right. And when the team depleted either Atx2 or hip, the embryos developed this distinct thorax cleft. They were missing their central bristles because the epithelial sheets just failed to fuse together.
Wow. So it proves this Atx2 hip JNK pathway is actually an ancestral physiological requirement for coordinated cell migration.
What's fascinating here is the sheer elegance of this post-transcriptional regulation. It's controlling the pathway not by flipping a switch on or off, but by changing how long the messenger RNA sticks around.
It's all about duration. Okay, time for a final synthesis recap before we look at the human pathology. Bullet 1. Atx2 stabilizes hipk mRNA by binding its 3' UTR. Bullet 2. This protective sleeve prevents degradation, leading to excess hip protein. Bullet 3. Excess hip hyperactivates the JNK cascade, which drives everything from normal morphogenetic movement to deadly tumor invasion.
Perfect.
Which brings us to the clinical frontier, because, Drosophila mechanics are elegant, but as a listener, you need to know if this protective plastic sleeve operates the same way in human cells, and what happens when it malfunctions.
Well, human genomics introduce a bit of an evolutionary complication here. While flies just have a single Atx2 gene, humans actually have two closely related paralogs. This is the result of some ancient gene duplication event. So we have ATXN2 and ATXN2L, which stands for Ataxin-2-like.
Okay, so the researchers shifted their assays to human HeLa cells to test both of these paralogs, and the functional divergence they discovered is pretty stark. When they overexpressed the sibling protein, ATXN2L, it robustly activated the exact same hipk JNK pathway they identified in the flies.
Right. It induced apoptosis in healthy HeLa cells, and crucially, it drastically upregulated cellular motility markers.
And they corroborated this in vitro data by mining massive human cancer transcriptomic data sets, right?
They did, specifically using the Cancer-C database. And what they found was a really compelling positive correlation between elevated ATXN2L expression and aggressive metastasis signatures in acute myeloid leukemia, or AML.
Specifically, high ATXN2L correlated heavily with EMT, the epithelial mesenchymal transition.
Which is such a critical factor in cancer.
It really is. EMT is this sinister biological mechanism where a static, surface-level epithelial cancer cell radically alters its entire cytoskeleton. It downregulates adhesion molecules like E-cadherin, so it separates from its neighbors, and it upregulates structural proteins like vimentin.
It essentially grows microscopic legs.
Exactly. It allows the cancer cell to break through the basement membrane and intravasate right into the bloodstream.
And the transcriptomic data heavily implies that ATXN2L is the human dispatcher driving that metastatic transition via JNK amplification. But honestly, the most revealing data point in the entire paper comes from how they tested the other human paralog, ATXN2.
Okay, here's where it gets really interesting, and we circle all the way back to our opening hook. Because remember, ATXN2 is the gene that, when mutated, causes spinocerebellar ataxia type 2 and is linked to ALS.
Right.
The mutation is a polyQ expansion. It's an abnormal repetition of CAG codons in the DNA. That results in a long, toxic string of glutamine amino acids in the synthesized protein.
And this causes the protein to misfold and basically aggregate in the cytoplasm of neurons.
Right. And the prevailing theory in neurology has largely been that these polyQ aggregates cause disease via a toxic gain of function, like the clumps physically damage the neuron.
But this paper's findings add a really critical layer of loss of function to the disease model. The researchers engineered a mutant polyQ expanded version of human ATXN2 containing 117 glutamine repeats, so mimicking a really severe disease state.
And what happened?
When they tested this 117Q mutant, it completely failed to activate JNK signaling.
Wait, really?
Completely failed.
So the expanded glutamine tract chemically breaks the protein's ability to stabilize the hip 3' UTR. The structural deformation prevents the dispatcher from binding the mRNA and putting on that protective plastic sleeve.
Exactly. And this raises an important question regarding evolutionary divergence. The data suggests that over millions of years, ATXN2L retained that ancient dominant role of regulating the hip JNK stress response, which makes it the primary driver of tumor invasion when it gets dysregulated.
So does this mean the human body has two slightly different dispatchers? And while one ATXN2 is famous for breaking down in the brain, the other one, ATXN2L, is quietly driving cancer metastasis in the rest of the body.
That is exactly what it means. ATXN2 evolved highly specialized distinct roles in human neuronal homeostasis. But when that specific polyQ mutation occurs, it doesn't just create toxic clumps. It fundamentally breaks ATXN2's ancestral stress signaling capability.
Wow.
Which likely contributes to neuronal death because the cell just loses its ability to properly manage physiological stress.
Okay, so what does this all mean? Like, for you listening, why should you care about fruit flies and RNA?
Right.
Well, if you're studying oncology or molecular biology, this paper just handed you a massive new therapeutic target.
Oh, absolutely massive. Targeting the JNK pathway directly to stop cancer metastasis has historically been a pharmaceutical nightmare.
All right, JNK inhibitors exist, but they're notoriously toxic because JNK is so fundamentally critical to normal tissue repair and immune function. You can't just carpet bomb the cell's main emergency response system without destroying the patient.
Precisely why identifying an upstream, highly specific regulatory node is so incredibly valuable. If pharmaceutical companies or structural biologists can design a small molecule inhibitor, or maybe an antisense oligonucleotide, that specifically targets ATXN2L.
Or uniquely disrupts its binding affinity for the hip 3-foot UTR.
Right. They could theoretically strip that protective plastic sleeve off the mRNA.
And by destabilizing that hip transcript, you lower the concentration of the hip kinase and gracefully turn down the volume of the JNK signal, but only in the tumor environment.
You throttle the metastatic EMT process without poisoning the patient's baseline JNK activity. It's a perfectly elegant post-transcriptional backdoor for cancer therapy.
It really is.
And if we connect this to the bigger picture, I think it forces a profound reevaluation of how we view the non-coding regions of our genome. I mean, we spent decades treating the 3-foot untranslated regions of mRNA as genetic exhaust, just useless tails trailing behind the important coding sequences.
Right, we literally called it junk RNA.
Exactly. But this paper proves that proteins like the Atx2 family are using these exact junk regions as hidden control dials. They hold the power to execute massive cellular decisions regarding life, death, and metastatic invasion.
That's wild to think about.
It leaves us with a really provocative question for the listener to ponder. If the 3-foot UTR of a single kinase transcript is secretly managing the metastatic fate of a cell, just waiting for the right RNA-binding protein to unlock it, how many other critical disease mechanisms are currently hiding in the untranslated, supposedly silent margins of our transcriptomes?
It's a humbling reminder of biological complexity, isn't it? We spend immense resources analyzing the genetic mutations that build a cellular metropolis, while completely ignoring the RNA dispatchers, quietly directing traffic in the margins. Until we realize, they were the ones holding the fate of the entire system in their hands all along.